Good evening to you from Christian Medical College, Vellore, the training desk of the COVID Command Center, to the 8th Pulse lecture on the laboratory diagnosis of COVID-19, the experience from CMC Vellore. The aim of the Pulse lectures is to provide up-to-date, accurate, and relevant information to inform practice both in CMC, in our mission hospital, and in the wider network across the country. We have been having lectures on a variety of topics, and this is the 8th in the series, the first one on the laboratory diagnosis. The good news from CMC is that the epidemic seems to be declining. The COVID wards are closing. And I'm going to start today's session with a few verses from a poem titled, "We Have Survived to Tell the Tale." The deluge has passed us, not left us. We have been shaken, but we are still standing. How to think what happened, how to remember, and how to bring to you who have not experienced it what we have been through. I don't know how we managed, I don't know how we survived. Through grit and purpose, never say die, not counting the cost of anything that was required. Key to it was administrative planning, without which we would not have been here to tell you the story, but more important, it was teamwork. We were united, we were one body, head, hands and feet, each with their different roles, each person gave their utmost, no one ever said no. Each person looked after the other, inquired about what was happening, going through for them. I'm going to introduce here a very important department in our COVID response, and that is the Department of Clinical Virology. At the beginning of the epidemic, I remember that we were not allowed to test, and we had to send samples to Chennai and to the government hospital. We were even not sure that we were seeing COVID-19. Since then, the Department of Clinical Virology has formed the laboratory backbone of the COVID-19 response, and without them we could not have looked after the 9,000 patients who have gone through our hospital, about half the patients from the Vellore district. And today we have two senior clinical virologists to share the experience with you. Dr. Mahesh Murthy, who has actually spearheaded the COVID testing from the lab, and Dr. Shoba Mammen, the head of Department of Clinical Virology.
Good evening, friends. Today's topic for the 8th COVID Pulse lecture is on lab diagnosis of COVID-19 and experience from CMC Vellore. A reliable lab diagnosis is essential for the early and the correct diagnosis of COVID-19 to identify new cases and monitoring treatment of confirmed cases. Accurate and fast test results may permit more efficient use of protective and isolation resources and allow for rapid therapeutic intervention and thereby prevention and control. With the onset of COVID pandemic, the Department of Clinical Virology in CMC Vellore started testing from the month of March 2020 and has tested approximately 35,000 plus samples, of which we had a positivity rate of 30.6. So our department was providing batch-wise testing 3 times daily with a turnaround time of the result within a period of 6 hours, and also a rapid result within 2 hours. So thank you for that. And I'm very happy to introduce my colleague, Dr. Mahesh Murthy, who is currently the professor in the Department of Clinical Virology. He's an eminent virologist and is primarily responsible for managing the flu pandemic in the year of 2009 to 2010 in our hospital. He has set up the lab diagnosis for flu, multiple respiratory viruses, and also the lab diagnosis of COVID-19, both PCR, screening, confirmatory, and also antibody testing. He's the principal investigator in many DBT-funded projects and has many national and international publications. So over to you, Mahesh. We look forward to an exciting lecture.
Thank you, Dr. Shoba, and thank you, Dr. Anand Zach, for those words. And I think it's very important that this topic be brought up, because we have been integral to the pandemic, and the response at CMC has hinged on what we were able to provide. And I think the key word that Dr. Anand mentioned was teamwork. And I think without that teamwork, even within the department, we would not have been able to manage this pandemic. At the outset, Dr. Anand Zach had suggested that there are a few key questions to address, and through the course of this talk, I will probably address all of these, and if there are any further questions besides this, you can always write in your chat and let me know if you want specific clarifications. I'll start with a little introduction about coronaviruses. They're a diverse family of viruses that infect many different species. The reservoir of coronaviruses is essentially in bats. There are 7 pathogenic species which have been known to cause either outbreaks of severe disease or cause mild disease. The 4 so-called seasonal coronaviruses are a cause of frequent illness both in young as well as adults as well as the elderly. The coronaviruses belong to the family Coronaviridae has several genera: alpha coronavirus, beta coronavirus, which mainly contain the human pathogens, and gamma and delta, which contain other pathogens of other birds and whales. Now, I'll move very quickly to SARS-CoV-2, which is the causative virus for this pandemic. It's a single-stranded RNA virus of about 29 kb. The key structural proteins of interest: the spike, the membrane, the envelope, and the nucleoprotein. Now, any lecture on laboratory diagnosis should have a slide which discusses the natural history of the disease, and it's important that any clinician who's advising a particular test also understands that just because you order a test doesn't mean it becomes positive or becomes negative, but you must understand that the natural history progresses in a certain manner, and you can find different assays that will help you clinch a diagnosis depending on what stage of the disease it is, whether it's early on in the illness. And in the case of COVID, we find that there is a pre-symptomatic phase, and at this stage, very clearly it's suggested that detection is very unlikely unless you specifically look for it. But after the symptom onset, you find that you will very likely get PCR positivity, and with time you find PCR positivity wanes and you get antibody detection. So it's important to know these features to choose the right test for diagnosis, a clinical diagnosis of COVID. So broadly, you can classify the laboratory diagnosis into either direct detection methods or indirect detection methods. So direct means you detect a part of the virus, one of its proteins, or the genome. And the gold standard and the most commonly used assay that has been used in this pandemic has been a PCR-based assays. And there's been a wide variety, which I will come discuss a little later in a few slides down the line. There's also antigen detection methods, either by ELISA or by rapid diagnostic tests, and sequencing of the viral genome, which has also happened quite a bit in this pandemic, and virus culture, which is restricted to certain higher containment facilities with post-culture confirmation. Indirect detection essentially is the detection of an immune response to the virus. It means that when you're exposed to this particular virus, your body's immune system mounts a response, and can either be the production of antibodies, IgG, IgM, or IgA, to the immunogenic proteins. And this can be detected by ELISA or other chemiluminescent methods, or a detection of a cellular response. So the guidelines for any diagnostic test is, it's important that for direct detection methods, the sample that you collect should be from the end organ or from the site of shedding. That's where you're likely to pick up any virus, pick up any either the virus or direct antigen. And it's also important that the method of collection be done correctly. And if there are, and that is one of the key factors that influences the sensitivity of the assays, because of the variability in the yield of the different specimens that are used for diagnosis of by PCR. Now, it's also important, as I mentioned earlier, what time of the illness, whether early or late, and depending on that, the modality you will use would be different. And we often ignore this, but the mode of transport, where cold chain is required to maintain the viability of the virus, to maintain, they tend to be labile. So especially in the borderline cases, the ones which are not so strong positive, you may actually get a false negative. So what are the specimens that you can collect? Now, any respiratory viral diagnosis requires a specimen. As I mentioned earlier, the direct methods require something where the end organ is affected, either the end organ in this case either be the lower respiratory tract or the upper respiratory tract. So all along the upper and the lower tract, any sample that is collected will serve for direct detection. And it's important that these respiratory specimens be placed in viral transport medium, which has protein stabilizers, which ensures the virus is preserved. There have been variations to the viral transport medium, which is the one used for the TrueNat assay, which is a decontamination medium where the virus is very quickly decontaminated and serves to help those who are transporting the specimen. Okay? Going back to lower respiratory specimens, it's important to have the bronchoalveolar lavage or the endotracheal aspirate, because these are specimens that can help in diagnosis of the severely ill. Okay, so which sample is recommended for diagnosis? The earliest recommendations that came, and there were preliminary studies that studied these and reported that the nasopharyngeal swab is the best in terms of giving you a better yield. But it is, if you see the procedure that's being done, it's quite a painful and distressing procedure for the patient, and compliance is not ensured at every instance. So there's a lot of variability in the specimen that is collected. So alternatives have been looked for, and nasal and throat swabs work just as well, but it depends on which stage of the illness it is. If it's early in the disease, there is actually no difference between a nasopharyngeal versus any of the other specimens. Saliva has been looked at as a specimen, as has sputum. Now, saliva, there's a special method which uses a PCR method without the need for extraction, so it's a more rapid method, and it's also self-collectible. So those are good options, especially when you don't want to have institutional collection. ETA and BAL are specimens from the lower respiratory tract. Okay. Our experience at CMC has been that we find that endotracheal aspirates don't seem to pass our quality check that we have for samples, and we find that the internal control, which is a control to say that the extraction was all good, seems to fail more often than not. So we usually have a protocol, which again I will discuss in the later slides, on how we deal with these situations. Okay. I'll move on to the basics of PCR. The basics. Now, all of you don't need an introduction to PCR, and I think it's been talked about quite a bit, but it's good to have a refresher. And so PCR is a method whereby there's an in vitro amplification of a specific target nucleic acid by the use of primers and by varying the temperature in 3 different stages: denaturation, annealing, and extension. At different stages of the PCR cycle, now all of these 3 stages together are called as a cycle. At different stages of the PCR cycle, different steps occur. So in denaturation, there is a denaturing of the template DNA, which is the DNA which you want to find out if it's positive or not. In the case of a virus, which is an RNA virus, you actually have a cDNA or a complementary DNA step. So you basically have a double-stranded DNA, And in the denaturation step, you have splitting of the template strand into two, they separate out. And then annealing is where the primers bind to the, if they are complementary, they will bind. And extension is the extension by use of a DNA polymerase. Now this is repeated cycle of after cycle, and you find that there is a increase in the specific product that is between these two primers. So the product, which is the forward and the reverse primer, binds. Basically, the forward primer binds to a region and the reverse primer binds downstream to it, and the region between these two primers is amplified, and it increases at the rate of 2 to the power of n, where n is the number of the cycle. Now, that's a basic PCR or a conventional PCR. When you throw in a Tachman probe, which is a fluorophore, which binds to a region between these two primers. So what that does is, when you have an extension, the Tach polymerase has an enzyme, the DNA polymerase has an activity where it cleaves anything that's in front of it. So when you have a probe that's bound to the region between the two primers, and the Tach DNA will cleave it off, and it separates the fluorophore from the quencher. Now the probe has two dyes. One is a dye that emits fluorescence, another one is the one that quenches it. So when they're in close proximity, there will be no fluorescence. The moment this cleavage happens, this fluorescent dye moves away from its quencher, and thereby there is a increase in fluorescence. Okay. Now this fluorescence, as I mentioned with the steps of the PCR, you find that with every cycle there is a 2 power n increase in the amount of the template of that particular region, which is specific to that virus you're looking for. Now, along with that, there is also an increase in the fluorescence, which happens where at every cycle there is an annealing step, and thereby fluorescence also increases. And this cyclical increase in fluorescence is detected as a sigmoidal curve. A CP, a crossing point, or a CT value, cycle threshold value, is the cycle number. As I said, PCRs are multiple cycles that are going on one after the other. The CT value is the number of the cycle at which the fluorescence that is being detected crosses a predetermined threshold. Now, when it crosses that, the sample is called as a positive. The advantage of using this kind of a fluorescence method is that if you know how much of input is there in the first place, and you make serial dilutions of it. So when I say how much is there, you make a DNA standard where a fixed amount of DNA is present, and you make serial dilutions of it. The samples that have a lower amount of the DNA will reach their CP crossing point, or the CT value will be higher. So if a sample is a strong positive, you will find that there is a low CT value, and if a sample is a lesser in amount, then you will find that there is a higher CT value. And there is a relationship between the concentration of DNA and the cycle number. Now, that is the basis for quantification in real-time PCR. You must remember that CT value is not an absolute, and you have to understand that for the same virus, for the same sample, different assays can give you a different CT value. It all depends on the dynamics of that, the way the primers bind and how the PCR reaction continues. Okay, so that enables quantification. Okay. Moving on to the real-time PCR assays for COVID. Now, this was a novel virus where there was it wasn't known what the content, we didn't even know that it was a coronavirus until the first sequencing results came. But it turns out that one of the assays that was used for surveillance in bats was actually able to pick up this assay, and that's the WHO E assay, the SARS-CoV-2 assay, which was able to pick up this particular virus. Since that initial stage, there have been multiple assays being developed, and they either target the E, the envelope, the nucleoprotein, or the S protein. Now, there have been different variations of it, and each agency, for example, the WHO recommends E and the RDRP, and the CDC recommends the N1 and the N2. Now, if you can see the figure here, you find that most of these assays, except the WHO RDRP assay, are fairly similar in their performance, and they tend to be just as sensitive. Okay? But you must remember that though they're highly specific, sensitivity depends on the virus load, the level of replication, and the amount of the way the RNA is isolated. Okay? There were also conventional PCRs, and they are specific for SARS-CoV-2. RDRP is the most common, though in real-time PCR we found that the RDRP was fairly insensitive. There are also pan-coronavirus PCRs which detect all known coronaviruses, and it requires sequencing to confirm the identity. Now, we at Virology have been exposed to a lot of commercial assays, and in fact the assay that we use most commonly, it forms the centerpiece for our diagnostics, is the Altona RealStar assay, which again uses two genes, the E and the S. So only if a sample is positive for both the genes is it considered positive. We also have some experience with the C gene Allplex, which uses 3 genes. There are cartridge-based nucleic acid amplification tests, which is what my colleague Dr. Shoba was mentioning about rapid detection, and we have assays being reported in less than 2 hours. So these are the Cepheid is the most commonly used system, and we use it routinely for solving discrepant results, but in addition we have also now begun offering it for patients on an emergency basis. There's also the TrueNat assay, which is a chip-based assay, which is based on the RDRP gene, but its sensitivity is a bit lesser than the other commercial real-time PCR assays. Now, we've also been exploring using a molecular point of care, the Abbott assay, the ID Now, which is very popular in the US, where it is said to give results in 13 minutes. We're exploring how we can use this, but you must remember that these are less sensitive than the benchmark, which is the proper real-time RT-PCR, which is usually the most sensitive. And among all of the assays, we find that they're fairly similar, but the CDC N1, N2 is more sensitive or slightly better than the others. Okay, so this is our reporting algorithm at CMC, and you see this red box over here. Find that most of our assays are reported as negative. Most of our samples are reported as negative and positive, simply because there are 2 genes in a single reaction, and we are able to report this. Now, sometimes we have some assays where, and it's happening more frequently now, and we don't really know the exact reason for it, but we find that though the IC or the internal control has passed, only one of the gene amplifies. So in order to resolve that, it only comes as a presumptive positive if one gene is amplified. So we usually resolve it by doing an additional assay, which is the Cepheid assay. And with this combination of assays, we've been pretty much 99% of our samples have got a conclusive result. And there are no very few samples where we've asked for repeat samples and tried to reconfirm the result. Antigen detection is something that when a virus is replicating in the respiratory tract, it produces some of its antigen, and that antigen is produced usually in very high amounts, and usually it's either the matrix or the nucleoprotein. And this has been seen very commonly used as a method for rapid point-of-care bedside diagnostics for flu and RSV. And the same has been employed, or it was attempted to do the same for SARS-CoV-2. There are issues where, as with any antigen detection assay, if the prevalence is less than 10%, you find that the accuracy indices are fairly poor, sensitivity is poor, though they may be really specific. It still doesn't serve. There are a large number of false negatives that will crop up, and that's the reason why it is not used and should not be used as a standalone assay for diagnosis. it cannot be used for diagnosis, but if the pretest probability is high and there's high community level transmission, then it can serve as a rapid method to characterize patients as either positive or negative. The recommendations by the WHO is that an assay of about 80% sensitivity and greater than 97% specificity should be used. And there are many, as I've said, along with PCR, there was many companies that came up with antigen detection kits as well, and everybody wanted to, because in the end it's an economic output, so they wanted a lot of kits to be evaluated, but we found that not all match this level of sensitivity or specificity. Okay? And in this scenario, when the WHO recommends that you can use it in a suspected outbreak when nucleic acid NAT tests are not available, and when there's widespread community transmission, early detection will enable the isolation and quarantine of individuals. It's not been recommended for testing of asymptomatic contacts, but the rationale here is that if you're asymptomatic and you have RNA, it's very likely that you are probably in the pre-symptomatic stage, or that it usually will be a fairly high amount, high amount of virus, so therefore it will be useful to use this device. Now, following on from the asymptomatics which I was telling you about, if you look at this, it's recommended that if you use a poorly sensitive assay fairly frequently at regular intervals, you're very likely to pick up an early you will very likely to pick up a case early on in the course of the illness than use a high sensitive assay which is used infrequently. So that's one of the rationales for suggesting that frequent antigen detections, antigen detection assays can be used as a method for quickly detecting and isolating cases. Okay. The other method that's I think probably the first time that we've had so many, it being so widespread, was the sequencing, and we found that within a very short period of time there are almost 100,000 genomes globally. Take, for example, if you compare it with influenza, there are only 20,000 genomes that happened during the 2009 pandemic. But with this, there's a massive number of genomes that were produced. In India, there's less than 0.05%, fairly infrequent, and it's restricted to certain centers that do the sequencing. The reason why this has been really successful is because there's been an NGS approach, and the method used is mainly to generate overlapping amplicons that cover the entire genome, and there are standard primer sets that allow you to do it, and all of the bioinformatics for it and for assembly of genomes is all almost automated, so therefore there's a large throughput in terms of number of genomes that are being generated. For example, the UK consortium for COVID genomics has something like 65,000 genomes on their own from the UK alone. So this is a large number of genomes. A feature of this pandemic was that there was very quickly there was a portal that was set up that was tracking the evolution of these viruses, and therefore it enabled very quickly to characterize how frequently or if at all these strains were diverging. Okay, so these strains are diverging, and we find that there was a lot of speculation about its origin, and we find that this virus, its closest relative is another sarbecovirus, which was probably diverged around 1948. Now, you must understand that they were very close together in 1948, but since then have diverged. Okay, so it's very difficult to make inferences based on a few nucleotides. So there's still the so-called origin or the closest relative of SARS-CoV-2 has still eluded us. It's not come from pangolins or snakes, or neither has it been engineered. There are different clades and many lineages that circulate globally, but the D416S spike mutant has been dominant, and there is evidence that it does tend to infect better, but we don't know, or studies haven't shown that it is epidemiologically fitter. Okay, I'll move on to the indirect methods for detection of SARS-CoV-2, and mainly we'll focus on antibody detection methods, and this essentially is the different methods like ELISA, CLIA, and other rapid methods that help us to detect IgG, IgM, or IgA antibodies. The key immunodominant proteins are the spike and the nucleoprotein, and in addition there is the spike, the receptor binding domain of the spike protein. Okay, so I'll go back to this natural history figure, and you find that it was, when looking at the antibody response, it was found that the antibody IgM and IgG almost co-occurred, and it was after 7 to 10 days of illness that the rise was pretty rapid, and therefore the utility of a antibody detection assay is not early on in the illness. So there is no point in collecting blood for serology and saying, is it IgM positive? It very likely that 7 days or 10 days into the after symptom onset is when it has utility. Okay, so as I explained, the receptor binding domain, which is the part that binds to the ACE2 receptor, is present on the spike protein. Okay, so both the receptor binding domain and the spike, as well as the nucleoprotein, are immunogenic, meaning that they tend to, antibodies are directed against them. Okay. There's a close relationship, as is seen in the phylogeny of it, of SARS-CoV-2 to SARS-CoV, and across all of the proteins, the N, the S, and the S1 and S2. The S1 and S2 are subunits of the S. Okay. The ELISA method essentially looks at detecting IgM, IgG, or IgA antibodies, and it's either an in-house format or a commercial assay. In addition, attempts have been made to look at peptide ELISAs. Now, peptide ELISAs are different in that it's not the whole protein that's used, but individual peptides that span, and they're usually overlapping peptides, which span the region of the antigen that we're looking at, in this case peptides of S and N. And it's found that in this one study that IgG antibody differentiates mild from moderate and severe. Found that mild illness tends to produce lesser antibody compared to severe illness, and the most severe are the ones that have much higher levels of IgG antibodies to both the S and the N. That said, if you look at both the IgM and IgN, you find that there is the N antibodies are slightly lesser than the S antibodies. So the automatic choice for an assay which can be used is the S or the SRBD, and that has formed the basis of a lot of assays, a lot of commercial assays as well. Okay, the chemiluminescent assays are a better way of doing it because it enables throughput. ELISAs have their own drawbacks where it takes a lot of hands-on time, and the amount of throughput and the duration it takes from beginning of the test to the result is fairly long. It can take about 4 to 6 hours, but the automated platforms enable the testing of a large number of samples, and also the time to results is fairly quick. And we have experience with the Siemens assay, which uses or detects antibodies binding to the S1 RBD, and the Roche and Abbott, which bind to the nucleocapsid. And we are still attempting to evaluate the liaison S1 S2 assay. It's a very short time to results. It's not known unless they explicitly mention it. We don't know which antibody is being detected. For example, the Siemens assay mentions it as a total antibody or IgG antibody as a separate kit, but the Roche only mentions antibody. So we're still in the dark. We don't know which antibody specifically we're looking at, but we're saying that there is positivity, there is reactivity. Kind of the gold standard test for detection of antibodies is the virus neutralization test. As for any virus, the ability of antibodies to bind to the receptor binding site, or the site that initiates a virus infection, essentially means that if you are able to demonstrate an antibody is binding to this site and preventing a infection of a cell culture system, you can say that it's neutralizing the infection, and that is thought to be a surrogate marker of protection, where it's protection from infection, but we do not know if it is protection from disease, and I will probably discuss a little bit about that down the line. Now, in the neutralization assay, what is used is a live virus that is grown in a BSL-3 setting, and it's reacted with antibodies from a patient sample or any sample. And if there is neutralization of infectivity, meaning that there is no virus CPE, it means that there is antibody present. If there is CPE, it means that antibody is absent. So the absence of CPE means that there is actually antibody, there is no neutralization. And a variant of this is either the plaque reduction neutralization assay, which enables counting, while the virus neutralization test requires a trained observer, and sometimes equivocal results may be missed. The plaque reduction neutralization assay is more, has a better visual readout, and therefore is more accurate. And as similar lines, the focus reduction neutralization test relies on fluorescence-based detection of growth of virus. Now, the end point of the neutralization test is that you're going to detect antibodies that have neutralizing capability, and therefore is probably going to be a key parameter which will be looked at for vaccine and also for antibody-based therapy. Now, recently it was reported that neutralizing antibodies are stable up to 6 months from the infection, even in a mild illness. Another way of looking at neutralizing antibodies is the pseudovirion assay. Now, I mentioned to you that the requirement for virus neutralization is to have a BSL-3 facility, and we don't have that many BSL-3 facilities, and therefore the best way to do it is to try and do it where there is no infectious risk to the handler, because you don't want to culture a priority pathogen and have lab infections. So therefore a BSL-2 option is always, it increases the biosafety. So what is done here is the spike protein of the SARS-CoV-2 virus is reacted with plasmids that encode for lentiviruses, either HIV or BSV. And what happens is, when they are co-transfected, they form, along with suitable fluorescent markers, they form specific lentiviruses. Now, lentiviruses are viruses that belong to the same family as HIV and HTLV. Now, these viruses, when they acquire or when they express a heterologous protein, in this case the SARS protein, they're called as pseudovirions, okay, or pseudotype virions, and these lentiviral particles can be used in an infection assay. Now, to infect SARS, the spike protein binds to the ACE2 receptor, so that when the ACE2 receptor is expressed on a cell, we are able to have a productive infection whereby the lentivirus, the pseudovirus, enters the cell through the ACE2 receptor. Now, specific antibodies which bind to the spike will prevent this particular process, and there is a loss of fluorescence. I forgot to mention that when the lentivirus enters the cell, it produces a fluorescence, and that fluorescence will be inhibited. So it's a very good and highly sensitive readout. There are other assays, the virus neutralization assay, surrogate virus neutralization assay, which essentially is the same thing as the pseudovirus assay, but it's done in an ELISA plate where the ACE2 receptor is bound to the plate, and the receptor binding domain, which is bound to a conjugate, is made to react with it. Antibodies that bind to the receptor binding domain prevent this reaction with the ACE2 receptor, which is coated on the plate, and therefore there is no colored reaction. There are alternatives where you can do a high throughput assay, where the virus neutralization assay, which we were talking about earlier, takes about 3 to 5 days, and sometimes even more. So a high throughput assay done where the results are available overnight is also an option, where you have a fluorescent virus that's used, and the readout is a fluorescence based readout. As with the antigen assays, the IgM, IgG antibody detection assays are also fairly commonly attempted to be introduced into the market, but we know from previous experience with respiratory viruses that their sensitivity is quite poor, and therefore it doesn't really help. And when there is a decision or a downstream decision to be made, then it doesn't help us in categorizing either way. Okay? Basically, what happens here in a antigen detection, in a IgM or IgG antibody detection rapid diagnostic test, is that the sample is captured on specific lines, both the IgM or IgG line, and with appropriate controls being in place, which are anti-species antibodies which capture the reagent. Now, when you find a colored line, and it indicates that there is that particular antibody, so a colored line at IgM indicates presence of IgM, and a test is valid if the control line lights up. Okay? And these are very rapid, meaning that they're called rapid diagnostic tests because the result is available within 15 minutes of adding the sample, and it requires, it mainly uses serum. Okay, I mentioned earlier that there is a difference in the titer of antibodies that are produced, and severe disease tends to produce higher levels of antibodies, both the spike nucleoprotein as well as neutralizing antibodies. But we do find that when you use ELISA as a measure for immunity, you find that 20% of samples which are positive by an RBD ELISA do not actually neutralize, and 25% of N antibodies do not correlate with or do not neutralize, they do not neutralize the virus. So it's important to not make these extrapolations that if you're positive by an RBD ELISA, that you are producing neutralizing antibodies. Saliva is another alternative for detection of antibodies, and you find that the IgG, it's similar to serum, so it's an alternative method for diagnosis of whenever serology is being used. And it was found that IgA is higher in acutely ill patients, but none of these assays are available routinely. Now a little bit about reinfections. I'm done with the antibody part of this, the talk. Now, we know reinfections are common for respiratory viruses, and when you describe the epidemiology of viruses in children, you find that viruses like RSV, PIV 1, 2, and 3 tend to reinfect frequently. The first episode is usually severe, and the ones that follow, usually within 3 to 6 months, are also, they may be symptomatic or they may not be symptomatic, but usually over the first 2 years of life, there is a decreasing severity of illness, and reinfections are common. And they've demonstrated this quite elegantly in multiple cohort studies. Recently there was a cohort study that described reinfections with seasonal coronaviruses, where they used antibody-based assays over 35 years. They had a sample collection from 1985 all the way through to 2010 and 2020, I think 2019. And they found that the reinfections were fairly frequent. The earliest reinfection with seasonal coronaviruses were 6 months, and the median time was 30 months. Now, this is important to consider because we're also talking about reinfections in SARS coronavirus 2. Now, reinfections are not as frequent, because if they were that frequent, we would probably be looking at many more infections, and possibly, we don't know, but there have been a couple of reports of them being more severe. But we have seen evidence that there is short-lived immunity in some cases, or immunity wanes really quickly, especially in the milder illness. So we don't know yet whether reinfections are an issue, but what is recommended now is that antibody positivity after 90 days from the original positivity should be considered as a reinfection, and appropriate tests should be done. So you have to document the fact that there were samples in between which were PCR negative, and most often that's not possible. And if you have seropositivity, initially you have to demonstrate the boosting of an immune response. Okay, so based on these, we find that after 2 weeks, the antibody assays are useful. They have a fairly high sensitivity and can be used, but they're recommended as per IDSA only in epidemiological studies, but can be used when there is a high clinical suspicions, where samples fit the clinical picture very well, but very late into the illness, and therefore the molecular tests are repeatedly negative. And this is useful both in serum as well as, for example, CSF, where you find that the manifestations can be very similar to the reported neurological manifestations of SARS-CoV-2. Now, you must remember that you should not use antibody assays as a determinant of immunity or risk of reinfection. And there are similar, so you cannot use, can't cohort people based on their antibody titers unless they are neutralizing antibody titers, and even those, though they last for up to 6 months, can wane, especially with milder infections. We find that the correlates of protection and correlates of progression are not very clear. We don't know what exactly is the correlate right now. There is a couple of reports which say that neutralizing antibody prevents infection, but we don't know how long it lasts and whether that can be a correlate of protection. Seroprevalence is what antibody assays are used for, and we find that there is a fair amount of false negatives as well as false positives occurring, and I think it's just a matter of time before the assays improve. It's just been 9 months into this pandemic, so as further iterations of these assays come out, you find that they will get better, and we will probably have better accuracy indices. Utility in RT-PCR negative cases is definitely there, but the problem is most of the assays that we use don't specify what antibody is being detected, so it could well just be a IgG antibody, which doesn't really help unless you do a paired serology. And there is definite utility in familial transmission studies, and it's been shown that superspreader events are the ones that contribute mainly to transmission, but within the household the attack rate is fairly high, so serology in familial transmission studies would probably be very useful. Up to 90 to 95% of adults produce neutralizing antibodies, and that's been reported recently. Okay, I won't go much into T-cell response because there's a fair amount that's come out, and there is a clear reports that describe immunity to SARS-CoV-2 by demonstrating activated CD4 cells to the different proteins of SARS-CoV-2. Now, what is interesting is that those who are unexposed, and this was found in 3 or 4 cohorts in many parts of the world, that people who were unexposed to SARS-CoV-2 or had no documented evidence of infection still showed reactivity to N peptides and other peptides from the other internal genes of SARS-CoV-2. So that's interesting, but that does not necessarily translate to any kind of protection. If anything, a T cell response may reduce severity of disease and does not mean that it will prevent infection. Okay, so sorry about the figure. I've drawn a dud here, but this was actually. I want to quickly go through the pandemic at CMC. Now, we've started testing, as my colleague Dr. Shoba mentioned, that it started in March. There were sporadic and referred cases, but from early June, we found a distinct peak, and we were going at 250 to 300 samples per day, and sometimes by the middle of June, we were at 2,000 samples per week, and the positivity rate initially started off at 10%, but then was consistently at 30 to 40%. And as Dr. Anand pointed out, that we are still standing and we're coming down. The positivity now, end of October, is about 20%, and it's going down even further. Children have been relatively spared in terms of numbers, and they haven't had severity, but you would have heard from the colleagues in pediatrics where they do describe severe illness among ICU patients in pediatrics as well. Okay. I want to very quickly go through the testing pipeline, and Dr. Shoba mentioned that the turnaround time of 6 to 7 hours, it's phenomenal that we are able to deliver results consistently day in and day out, almost 7 days of the week, with a turnaround time of 6 to 7 hours. So this flowchart here kind of demonstrates how much of effort goes into each of these different processes. I won't go into too much detail about it, but I have to say that without the efforts of the individuals in the department, the technicians and the AROs, and also the additional help that was given to us from all the other departments that provided us support, was the only way that we could have managed this level of this turnaround time. And a key help, or a big help with the reporting, was the fact that we had an online system, and we must thank CHIPS for this, for developing this fairly quickly. And now it has become so well streamlined, and we are very easily able to report a large number of tests in a fairly short amount of time. As I said, it's only 15 minutes for reporting once the assay is done. Okay. This is the algorithm that we follow. I think it's a repeat. I've explained this before. Okay. I think I've pretty much covered all of the questions that Dr. Anand has posed and said these are things that may be of interest, and I'm happy to take any further questions.
Dr. Mahesh, we have a few questions. First question, when should we order a repeat test if the PCR is negative but the pre-test probability is high?
So usually it's suggested that you wait for 2 to 3 days at least. Now, we have had reports, and this was though not that much from CMC, but you find that there is a variability in PCR positivity as well, where you have patients becoming PCR positive and then negative and then coming back up again. So it all depends on, if your pretest probability is high and your clinical suspicion is high, you're likely to pick it up. But you also need to consider the duration of illness and keep in mind the natural history of the disease, and you may be at a stage where the RNA has actually gone below the detection limit, and therefore you're getting it as negative. So it's probably a good idea to look at antibody-based assays for diagnosis in these cases.
Currently, what is the next question? Currently, what is the specificity and sensitivity of the RT-PCR test?
Okay, So if you have an ideal situation, your sensitivity and specificity is 100, okay, but you don't. And as I mentioned, the specificity is fairly high. It's probably 99.9, though there are a few instances of us having picked up some noise here and there, but that's part and parcel when you do a large, high level of testing. It's probably 99.9 in terms of the specificity, but sensitivity varies, and the sensitivity is dependent on the site of sampling and the method of sampling and what stage of the disease it is, whether it's early in the illness or late in the illness. So sensitivity has been reported to, if you have a high positive, if you're, say, if the virus is a fairly high amount of virus is present, you find that it's a very sensitive method. And that's also one of the drawbacks of using a real-time PCR, is that it's probably too sensitive, and it cannot be used as a measure of saying clearance of the virus, because you have PCR positivity that goes on, and we've had many instances of positivity going on many months. So that's a problem as well.
Next question, do we know frequency of antibodies against OC43, HKU1, 229E and NL63 in our population?
We don't know, but we have initiated cohort studies that will help us to find that, and this is a collaboration with Department of ID, and we will be able to see population level, at least have an idea as to how much is there. But going by epidemiology, you expect that there will be a high amount, because these are viruses that usually infect early in life. So even if you have reinfections, which was documented very elegantly in that paper, even if you have reinfections, you still will have some level of antibodies. And it's the assay that you look at, which depends on which protein you're looking at, either the N or the S, that will help us to determine. Long story short, we don't know yet, and we don't have information in CMC.
Next question, in 3-gene detection, if ORF is positive and E and N gene negative, how do we report it?
Okay, when you have a situation where you have 3 genes, now there's a problem with having 3 genes in a PCR, because it's 3 genes plus an additional internal control, so that's 4 genes. And the way PCRs are designed, the fact that you put all of these reagents into one tube makes it, one of them is usually out competing the other for all the reagents in the tube, so therefore you find that it's actually a false negative. The best option, if you have a situation where you have only ORF1 positive, that's considered as a screening assay, and you try and repeat it with an additional assay, and which is the methodology that we use in CMC, where we do an initial assay. If there is 1 gene positive, we try and resolve it by doing an additional assay, and in this case it's the Cepheid assay that we use. You will have to retest.
This is a pediatric question. As children have more PIMS TS, we wish antibody testing to be available readily.
Is that a question?
Question or a comment, I think.
Okay. Yeah, we should have that now. As far as we know, ICMR doesn't allow you to conduct or offer tests as a service, and I believe that's going to change. We hope that it will change fairly quickly, but it is useful to do that, and it can be done on a research mode where you try and find out, and that is something that we have also been working with the Department of Pediatrics at CMC to offer this as a test for diagnosis on a research mode.
Any more questions?
No question.
Do we need to have exponential line always to report it as positive? That means in PCR.
Okay, so if you want to, now it doesn't usually happen like that. When you have weak positives, you find that there isn't an exponential amplification. There's just a hint of a line that we are then stuck with we don't want to sit on the fence. We want to be sure that is it truly positive. Now, when you have these weak positives, that's usually in the range of CT38 and above, that you get these non-exponential, just a straight line that just seems to suggest an increase. Now, that's a problem because repeat testing will make it negative. When you re-extract the sample and redo the test, it will be negative. When you ask for a repeat sample, which is usually a day or two later, you will again have a negative result because it's, as you know, the natural history, the virus is going down, it's going to go down further. So therefore, it will actually be negative. So it's a decision to make, and that decision is what you do with that original sample. You test it once, and if it is just an uptake, you check and see if it is a weak positive in 2 or more genes, then you can report these as positive.
Is WGS test of choice if index of suspicion is high and antibody and PCR are negative? WGS?
Okay, so whole genome sequencing is dependent on us detecting the virus. Okay, you need to have a certain amount, and it is nowhere as sensitive as a real-time PCR. So, for example, we have a kind of a cutoff value of CT32, below which you may get decent reads. You may not get whole genomes, okay, but CT28 is when you will get whole genomes. Now, you could use it, and that's been suggested as a method whereby, if you use deep sequencing, you'll be able to find out if there is at least, one particular read or less than 50 reads that map to this particular virus. But it's too expensive for you to do it for individual samples, and the throughput is going to be very, very low. If you have a high index of suspicion, I think the way to go is to go either, if PCR is negative, antibody is negative, repeat the antibody in a few days, usually 2 to 3 weeks, and then we can have, we may find that there is seroconversion.
So I want to thank Dr. Shoba and Dr. Mahesh for sharing the experience of setting up this the lab services to support the COVID response at CMC, and sharing all their experience that has come out of the work that has been done. I think as we see the epidemic declining here, the challenges will be for other waves that we get and what's going to come up in the next year, and how to maintain the kind of testing that will be required in the coming period of time. So we wish the department well, and we look for their continued support as clinicians for our work.